Where's the... Okay. Oh, there's... All right. Let's see, we've got 4 more minutes, people to dwindle in. I could like explain things, but I just don't want to waste my breath saying it 50 times over and over again. So we're gonna wait. Okay. coming in, join in on this Kahoot, you go to kahoot.it. You're going to the website. Otherwise, I highly recommend if you're going to be using your phone for these or your tablet that you download the app, which is going to be faster. And it'll prompt you for a code. That code at the top is the one that you'll use to start. You can also use the QR code. It'll just take you right to it. We'll use these every single day at the beginning of class. This is how I'm taking attendance, and attendance is for credit in this class to make it like attendance that's actually worse, like you did a thing. That's why we have the quizzes associated with it. Like I said, if you weren't in here previously, this is participation based. So even though every time that we have class, at the beginning of class, we'll have a Kahoot quiz that's 10 questions. It's going to review over the previous chapter. Today we don't have one, so it's just questions about like when you last took chemistry and some basic questions like that. But it's even if you get every single question wrong, you're going to get credit as long as you participated. But it's how I'm going to keep attendance. Having said that, I am not like super picky about you being here exactly on time. Like I'd like to get started with these quizzes typically within like the first 5 minutes. Like I'll put it up at like, you know, a little before 5:45 and at like 5:50, I will usually want to have started the quiz by then. However, if you come in late-ish and we're still doing the quiz, as long as you have logged in at least once under that number, I'll have counted you as having participated, okay? And it's gonna ask you for your email address that, if you guys haven't logged in yet, it's talking about your OK State email address. If you did sign in with the personal email address, as long as it has some aspect of your first or last name in it, that's what I'm gonna be using to make sure that I count you as present, okay? If you made up a username, some of you guys are putting your names in there, and that's great. That helps me figure it out and all that. In the future, if it lets you change your username, if you don't want to use an identifying username, that's fine, as long as you put in your actual email address behind that. You know what I mean? So, so whatever. It's not going to show that you put this exact answer, right? But it will put the top 3 people on a podium. So yeah, we'll do one of these at the beginning of every class. I usually want to start them within the first 5 minutes. And if you come in late and we're still doing it, just try to get at least a question or 2 in. If you come in late and you missed it completely, there's a paper up here every single day. It says lecture late arrivals. I also have them for lab. We'll talk about what that means for lab when we get into the lab. But if you come in late for the lecture, but you're still going to be here for most of the lecture, go ahead and sign in your name and the date in the appropriate spots. And then it is your responsibility to do the quiz that you missed that day. Once we get going with the chapters, those chapter quizzes, if you've taken a look on Canvas, we have, you know, optional Kahoot links. It's the same quiz. Okay, so we're not doing chapter 1 and 3 today. We're doing intro. The next time we come in, we'll do chapter 1 and 3 when we start, and then lecture. Does that make sense? So it's review is what it's meant to be. So if you were to come in late on Thursday and miss the quiz, you know you would have to go find chapter 1 and 3. Take a picture of your final screen. Again, I don't care what score you did. Just do it, right? Take a picture of that final screen that kind of prompts and email it to me. That prompts me to go ahead and look up and make sure that you did do it. You signed in on the sheet. That's another way to double check it and make sure that you get your credit for participation. Okay. That doesn't mean come in obviously late at the end of class. There's not enough people in this class, obviously, for me not to start noticing who that would be. Not that I would ever accuse you guys of trying to do something like that, but to protect those of you who do try to be here on time and regularly and everything like that, I don't want to violate your efforts and then let somebody come in the last 5 minutes of class and get credit for participating, right? That's the idea. So that's the layout for the lecture, what we'll have every day. We'll try to start Kahoots within the first 5 minutes of class, like I said, for participation credit. And we'll, in the beginning, probably until about February-ish at least, I'm going to go over those questions like normal boring. Like we'll go over them question by question and I'll discuss them and everything. If I feel like you guys are getting it and you're getting the whole point of the Kahoot, then we'll probably just switch to games. If you haven't played the games on Kahoot, they're pretty fun and they'll kind of repeated, like introduce new questions repeatedly and make it a little more fun reviewing through those chapters. But we'll kind of see how you guys feel about that after the first month of the boring way. I'll say boring, but the methodical way. Anyways, that's the Kahoot part. The second part, we will jump right into whatever the lecture is for the day. So today we will be lecturing over chapters 1 and 3, kind of combined. It's just intro kind of stuff. What is microbiology and what are some of the basic tools, generally speaking, that we use for microbiology. Then we will, at the end of every lecture, there are 5 questions tacked on to the end of the slides. They're already posted on Canvas. Those slides that I have on the PDF is accurate. Those are up to date in the slides that I'm actually using for the class. So at the end, there are 5 questions, okay? I will ask those to you. I am calling people's names out in the class and you will answer in front of everybody, okay? We're adults and we should be able to discuss this stuff as adults. So if you're someone who's nervous about talking about stuff like that, first of all, I'm going to tell you every day who I'm going to call out, okay? You get to know ahead of time. Number 1, that's that. Number 2, we're going to go in order. So it looks like from top to bottom, and I literally have like a dice app on my phone and I just pull up a number, okay? It's a 30-sided die. We only have 19 people in this class. I just use the 30 for everybody because most of my other classes have 24 and that's like the one that accommodates everybody. So if I roll anywhere above the number of students in a class, once per class, if that happens, I will cover one of the questions, okay? The other questions will be on you guys. So it looks like our first question that will come up, and I don't remember what it is. We'll get to it. You're welcome to always look ahead and prep for your question if you want to. But Lakita will be our first. Is Lakita here today? Yes. Okay. You'll be our first victim today. So I want to be clear on this. A lot of the questions, I feel like they're pretty just like basic, simple, kind of big kind of concept type of stuff. It wasn't something that was like some obscure random fact out of my butt, right? So it's typically something that I would expect you guys to get big picture wise. And usually most of the time has a definitive answer that you would be able to find in the notes. Sometimes it'll be kind of a vaguer question, and sometimes that's not intentional. It's just I don't always see it as vague, and then you guys will get to it, and I'll be like, oh, that was my fault. I didn't see it that way. Anyways, the whole point of these questions is not even necessarily that you get it right. It's that you're talking about the material that we talk about today. So if I ask you a question like, what was that molecule that makes up bacterial cell walls? The answer to that, by the way, is peptidoglycan, and we'll talk about that in chapter 4. But let's say you didn't know that, right? And you wouldn't know that today. And you remember we talking about it, and you're sure you could find it in your notes. But you're like, I remember that it had to do with like sugars linked together. Even if you said that, A+, man, you were listening and you're paying attention. Like you were following what I was talking about. You had an idea of what the answer would be. You get what I'm saying? Like that's what it's about. Engage with the content, not necessarily even being right. If you had like said, well, I remember we talked about something having phosphate groups and a negative charge, I would say, well, that actually was the membrane, not the wall, but you were still paying attention. It just didn't apply. So that's what I'm looking for, engagement. Those questions that are at the end, that's the number 3 up there on that board, are extra credit stuff. So if I call on you and you answer your question within the realm of what we were speaking about, like I said, doesn't have to be exactly correct, then I'll count that as, hey, you're engaging in the class, and that counts. Everybody gets 2 passes a semester. I doubt that you'll need them, and I feel like that's a pretty generous give whenever we have enough people in the class. I'm not calling on you every day, right? But hey, maybe it's not your day, or you just don't want to mess with it. So you can pass that day. Every day it's 2. If I call on you a 3rd time, and you want to pass a 3rd time, you relinquish your extra credit. So that's how that game's going to go. So by calling you and you're trying to participate, as long as you keep up with that, you get 1% added on to your final overall grade. That could be enough, right? That could be enough to put you into the next letter grade. We're allowed to get up to 3% added on. So 1% is going to come from. Another percent comes from completing everything through the course with no zeros. So as long as you complete it, like even if you get to the end of the semester, you're like, shit, I didn't do this quiz. Just knock out a question, man, and get at least one of them right, and you're good to go. Like, just make sure you have a non-zero grade for everything. Automatically, you get another 1% tacked on your grade. That's because you were a hard worker and you tried, man, right? Good job. The other 1% is for completing the course evaluations at the end of the semester, pretty much. Okay. They're not too hard on the extra credit, just basically like showing up and participating. Okay. Lucky you guys. I feel like getting called out is one thing to still talk about in front of class. I'm sure you're picking up on like I'm a pretty casual person, and I feel like my style of teaching, especially when we get into the lab, it's more like discussion, like casual discussion type of a deal on level type stuff. And you guys will get used to that, and you'll be more open to talking, and you'll start to realize that, hey, you're not the only one that's answering things quote unquote wrong, that as long as we're all having discussions about these topics, we're all learning from it, right? So that's way more important to me in the learning process of all this. Now, we want to keep learning open. We want to keep learning open, and I want you guys to feel like it's a safe space to, you know, to pause it and guess and feel out the information while we're in the lecture environment, while we're doing the Kahoots where there's no danger in getting anything wrong, or the extra credit where you're listening, but you're not exactly sure what the exact right answer is, but you're willing to participate in the discussion about it. Even if I ask you one of those questions and you're like, you have a question about it, but the question is applicable to kind of, you know, feed into the discussion about it, that's what I'm looking for, right, is engagement. Anyways, and then if we have time, again, if we have time for you to answer the questions. Today we have 2 chapters we're covering. So far in my other courses, I haven't gotten to the questions. So I don't want to leave you out though, because the questions might have information that you might like to review. So what we do, if I get to the questions and I don't have time to let you guys kind of sift through it, I will answer them. I will go through them all and I'll just be like, this question asks this. The answer is that, and blah, blah, blah. So that you guys can still pick up on that information and gain from it. I'm hoping that when we get into the single chapter stuff, you guys know that the first day is always one of those days where I'll give a little bit of intro. So that's all the intro pretty much for the course. And then if we have time, a relevancy video. Those are also posted on Canvas. Again, not required viewing. If we get to a relevancy video in like a chapter lecture, like today we probably won't, but if tomorrow we do, we get to our relevancy video and I start playing it, I will not be offended if you go out to have a drink or smoke or whatever it is that you do in your time between here and lab. I don't care. So if you feel like, you know, oh, relevancy video, great, like I don't care, but I'm going to play it if we have time, right? So if you want to take that time to go to the bathroom or whatever, I'm not going to be offended, okay? So FYI. But it's something to do in between the lecture and the lab that kind of ties things together and puts things in perspective. And again, those are always available on Canvas. If we don't get to them, you're free to view them on your own time. You're not going to get tested over them or anything. They're just to tie the concept in. So that's the lectures and what to expect on a daily basis and kind of what we're going to be doing in the lecture. For whatever, for the lab stuff, we're going to do lab stuff. Talk about that stuff on Thursday. I don't want to get too much into it. So we'll go over the lab stuff as well as like the Canvas structure and what you have to do and how you might want to use the extra stuff that's on Canvas. We've looked at that main like must read announcement that kind of outlines like the things that you have to do, have those little greater than signs on them to help you find that stuff in the modules if you're a module person. Also, obviously, the to-do list is over there on the right that just outlines everything you have to turn in on time if you haven't figured that out. And every Friday at 8 a.m., you will get an email from me telling you what you must have turned in by midnight that night, every Friday. Okay. So most of our assignments are due by Friday at midnight for the week. So everything we've done that week, your chapter quizzes and your post-lab quizzes, and making sure you submit your lab worksheets. Those things are all due by midnight. This is the week that we do them. Okay, Friday, the very last weekday. So you'll submit those. By then, I will absolutely send out a reminder every single Friday at 8 a.m. saying, be sure you turn all these things in this week by midnight. So this Friday, today, by midnight, so that you guys don't get too behind on that. Because I know you have lives outside of this and you might get sidetracked. So there you have a list just right there. You can click through and make sure you knock it all out. Okay. Friday. Yep. I don't do Sunday because I like to keep the routine of Friday, because once we get to the exams and once we get to, y'all are going to put it off till Sunday at midnight whenever we have the exam like the next day. And so I like to have it all one date for all of my sections, whether they're Monday, Wednesday, Thursday, Tuesday, whatever day the week you have the class. So it's all due Friday at midnight. Most of this stuff, like the in-lab worksheets, y'all taking pictures of it in the lab. There's no doing that outside of the lab. All you have to do is make sure it's uploaded Friday. There's no way to do it outside of the lab. So those things will be in the lab. We'll take pictures to sure it's uploaded by then. The quizzes, they're timed, and you get one chance. Okay, one opportunity to get them. So once you start them, please sit down and make sure you have the time to do them. It's not lockdown browser. You can Google it. You can AI. You can use your friends. You can all sit down and do it all together if you want to. I don't care. but it is time. So that makes sure that you guys are ready to take them. You've done some kind of familiarization with the content. And the 15 minutes take 10 multiple choice questions. Multiple choice or true false. Okay. 10 maximum. Some of them are 5. Okay. And we'll go over the details of some more of that stuff whenever we get to the lab, quote unquote. Thursday's lab is literally just me talking about the class, as well as some note-taking skills. Even my best students, y'all know Becky Dye. Anybody in here know Becky? Know Becky, then you know what I'm talking about, okay? You guys ever met one of those students that just is like does so well and like just like excels above everybody else? And of course they're going to do well in class or whatever like that. We've all met somebody like maybe you are that person. Maybe everybody in here is that person. I don't know. But even some of my best students, when I looked at their note-taking practices, y'all suck at note-taking. And you might know it. You probably do, actually. And you're like probably seeing like, yeah, I noticed I took physiology. I'm aware, like I'm not really sure how to, break down the material and get, the info I need for the test and whatever. That, guess whose fault that is? Not yours, right? That's everybody before you. That was your teacher before you that probably didn't set you up for success. And didn't take it upon themselves to try to do that. I don't have all the answers and I can't solve that within 16 weeks, but I'm going to do my best to try to help you guys along with that. And for those of you who do want to improve on that stuff, we're going to try to help you guys with that. Okay. We're going to talk about a little bit of that stuff on Thursday too, in addition to the core stuff. And obviously I have that module on Canvas for you guys to peruse. Note taking is a kind of figure out your own story kind of a deal, unfortunately, and studying is too. Flashcards are great for content memorization, but they don't actually put you in scenarios to apply. I'm sure you guys noticed that too. Yeah, you can pick out a term and what words they relate to, but they don't actually help you if I put you in a scenario of now on my test, where you guys are going to get questions of like a 52-year-old male comes into your ER with a fever and a rash. He seems to be sensitive to light, and he says that he hasn't had any respiratory symptoms and blah, blah, blah. Which of the following things do you think could be wrong with him, that sort of stuff, right? Sometimes in the beginning, that stuff can be overwhelming because I will give you more information than you need to answer the question. Absolutely, I will. Because guess what? How many of you guys are going into nursing? Please raise your hand. Don't be shy, right? How many of you guys are going into vet? Please raise your hand. Anybody going into dental? Anybody? Okay, those of you who didn't raise your hand, raise your hand. Where are you going? Yeah, trying med school. Yeah. So same kind of deal, right? Healthcare kind of field, right? So you get it. So those of you guys that are going to healthcare, whether it's human or animal, do you think that they're going to just give you the information you need to solve your problems? Absolutely not. Also, you guys have probably heard this is a challenging course. I get a pretty good, rep for how I teach, but it's a challenging course. I can't help that. It's just a lot of new content, and you guys are usually not prepped for biology stuff. That's also not my fault. Believe me, we've argued and argued that you guys need to have bio before you come here, and people in the future will have to have bio and finally won that battle. But y'all are suffering for that. So I do my best to try to prep you for that, but it would help, right? To have a little bit of primer for something. You'll agree once you get into the course. But that would have been, I hate to say it. I'm not trying to be rude, I'm just being transparent here, right? So anyways, having said all of that stuff, do you think if you guys go to someone who is in the nursing program now, find AJ, we know AJ, y'all don't know some people, you will probably when you get into nursing. People who are in like their, past their first year of nursing, right? Go ask them what the hardest course was that they ever took so far. Do you think they're gonna say micro or physio? You think it gets easier after that? It does not. And that's not me being nasty. That's me being real. And so I want to prep you guys for that. I'm not going to be like, oh, micro's so hard, and then you guys go on thinking it's going to get easier from here. That's why I want to prep you for the future. More important is that you guys be prepped for your success in the future than you carry on freaking micro. You guys can look this stuff in and put a book if you need to later on. Now, at least you'll know how to look up faster and where to look and what kind of details are important or whatever. That's the whole point of this, right? However, having said that, at the same time, my tests are not open notes. That's for babies. This is you come in my class and you're meant to learn microbiology, especially if you're going into med school. That shit don't fly. Okay, you're expected to learn this material. You're taking this. I'm expected to teach this class as an exchange course. Like if you were to take this microbiology course at the equivalent, if you took it at like OSU or OU or something, I have to teach it that way. So I expect you guys to learn the material. I do focus way more on clinical stuff than on like, I don't care about environmental micro or anything like that, okay? We're gonna focus on the clinical side of things. So it should apply, and it should be interesting. And I think you'll be surprised what you don't know about medicine, honestly, about the illness side of things, the contagious side. So honestly, you'll probably learn something today about that. So just keep all that in mind, keep an open mind, be ready that it's going to be work. This is not an easy class, it's a 2000 level class. So it's going to be some work, but also if you are willing to put in your end of it, then I'm always going to be here to support you with whatever it is you need. Right? So if you feel like you're struggling and you're not talking to me about how you're struggling and you need help, I'm not going to know. And if you're sitting there with a C, I'm just going to assume you're okay with your C if you're not coming to me asking for help. Okay? I'm here to help you every step of the way. I have so many freaking resources on that website, probably blowing your mind. You don't know where to start. You don't need all of it. Pick and choose the ones you like. Try some stuff out. I have those freaking chapter homeworks that I know everybody loves so much from McGraw-Hill that I don't assign for credit because I think they're boring. And I think that they're kind of a waste of time when they're assigned to you. However, you use one of those McGraw-Hill chapter things that you click through until you get 100 out of 100, mastery or whatever. It's pretty good primer for the lectures. It introduces you to the topics and without you having to weigh your grade on it. You don't even have to finish it if you don't want to, but it gets you to reading a little bit into it. Come to the lecture, and then all of a sudden the things I'm saying are it make more sense to you. Like I said, this is going to be a lot of new info, okay? I'm here to support you, but reach out to me if you need help, yeah? Talk more about the course on Thursday. For now, when we get to the end, if we have time, and like I said, we probably won't, it will be Lakita, then William, then me, then Aileen, and then, is it Aileen? Eileen, okay, thank you. And then Gabrielle, okay? So that'll be the order of the questions as well at the end. So if you guys are looking at the lecture, you can follow along and prep for your question. So like I said, this is the Kahoot. We'll do them every day. They do count for attendance. If you guys also, I know we already said, if you come in late, you can join in on the Kahoot, or if you miss it completely, sign in. And then be sure you do the appropriate Kahoot that you missed. Email me the screenshot and I'll give you credit. Well, if you don't want to come to lecture, if you're just one of those people that does better chilling at home watching the lecture, remember you have to be here for lab. That's just the bottom line, okay? But if you'd rather go sit in a dark hallway and watch the lecture, if you'd rather go sit at home and then meet us up for lab, if you'd rather sit in your car and watch the lecture, I don't care what you do, okay? You can join in on Zoom. We already have one participant with us on Zoom, and I can see who joined. And they'll be able to join in on the Kahoot and still get credit for participating. Sit in, watch the lecture. It's the same darn thing. So if you decide that that's something you want to do, you're going to be here for the lab anyway. You have to be. We don't have lab today. So on days where we don't have lab, maybe, yeah, opt out. That's fine. You still can get credit. Capiche? Sound good? All right, everybody good? We sign in? I'm going to go ahead and start. Oh, I'm gonna turn this light off. I don't like it when people throw lights on. All right, when did you last take college chemistry? There are some choices, so be sure you choose the one that applies to you. Like I said, like if you come in and we're already going, we're already cranking on these Kahoot quizzes. The code is down here at the bottom, so you can just go to kahoot.it and type that in. As long as you have logged into the Kahoot quiz, at least at some point before it's done, it'll count. I hope you heard me back there. Okay, when you get situated, go to kahoot.it and then plug in this code down here, 281865. If you can't read it, you can always holler at me and I'll tell you what it is. I know it's a big room and it's a tiny screen comparatively at the back. All right. Oh yeah, you guys, I'm sure you saw, most people did take it in the last year, even if you didn't. We'll talk about it in a second. But this is asking, I don't know why this has to be here. About biology, college level biology, not GOB. There we go. All righty. So have you guys taken college level biology? A lot of you guys have taken anatomy and physio, or one or the other. Maybe you took some other biology level thing. Most of the people will not have taken true college biology. Number 5 is actually pretty good, and AP bio does count if you did do that. This is obviously it's microbiology, but we're going to be talking about a lot of biology in this course, so it's nice to have that primer. And it will be a rare day that we're not having chemistry in this course. Okay? I don't do math. I'm sorry. We might double things in this class. We might multiply by 10. Okay. So you will absolutely need those skills when you get into nursing. This is not the class that's going to test those skills. More or less, I'm more interested in concepts. So we'll talk a lot about how things, we're going to have pH question here in a minute, so get ready, but concepts behind things like pH or hydrogen bonds. And we're going to have a chemistry, that's what chapter 2 is. Chapter 2 is a chemistry like review chapter. So lean into that. If you have any weak areas in like the organic and biochem side of chemistry, there's a lot of that in this course. I mean, it's like every day. Yes. Yes, exams will be, yes. Yeah, they'll be in person. You'll bring your laptop or your tablet to class and we'll do it on lockdown browser. Anybody in here have Dr. Rhodes? Yeah, did you guys, because you did that in his class too, right? You would bring your laptops and do like a lockdown browser situation. And you've probably done that in other classes too. That's what we'll do in my class. It's just easy for me to monitor. I don't have a lot of respect for like ProctorU and that stuff. Also, it makes people uncomfortable. Yeah. Stressful, right? I think, I mean, I don't know if anybody else does it. I know that Dr. Shearer leans into the lockdown version of it. It's like AI. Yeah.
It was on my birthday too, so I took a test on my birthday. When I showed him my ID, he's like, "Oh, it's your birthday."
And you're like, I don't know you. Oh my God. This is what you need on your exam day, right? Yes, what's up? Okay, it might be because we're on the middle of a thing. So let's see if it pops up when we move to the next question. Try refresh it. And then let me know if you can get it to work or not. We have a couple more questions. What is pH? As long as we get you in on one, you'll be okay. 281865. All right, what is pH? Pick the one that's the best answer. Oh my gosh, if you guys love a question, hopefully we can get it to work in the future. If I don't, what's your name? Okay, Desiree. If you can't get it to log in before the last question, come sign in on this sheet up here, and so that I can at least get you for credit for showing up. Okay. All right, pH. pH. This is one of those questions where you want to pick the best answer. Most of my questions on my exam aren't going to be like this. There's going to be an answer that is the right answer. There's not going to be like a which one's the best answer and they're all right or something. But when you guys get into nursing, you better believe almost every single question you're going to see on your test is going to be what's the best right answer, and they're all right. I'm not kidding. Okay, and y'all have to get at least a 75 on that. You can't pass with just a C. So here's the real answer for pH. It's a measure of hydrogen ion concentration. Yeah, are we looking at acids and bases? Absolutely. But pH isn't really a measure of how much acid is in something. It's a measure of the hydrogen ion concentration, right? That's a more correct way to look at it. We're going to talk about pH. Don't worry, there's a refresher for that in chapter 2. It does matter a lot for what we're going to learn. All right, proteins. Is this true or false? Are they a complex molecules comprised of saccharides? I'm not judging, really. Because again, this is stuff we're going to talk about in chapter 2, but it just gives me a sense of you guys are thinking about this stuff. If you guys took chemistry, you probably talked about proteins. Proteins are made up of amino acids, right? So we look at a saccharide. Have you guys ever heard the word saccharin? Like somebody had a saccharin personality or they had a saccharin smile. It means sweet. So what? Sweet? Sugars. What are sugars? Carbohydrates. Even in your macros in your food, are carbohydrates the same thing as proteins? No. Okay. So even if you have to break down with that type of a thinking, that's okay. And like I said, we're going to talk about this in chapter 2. It's okay if you didn't get it right. All right, true or false? Bacteria can be infected by viruses. Did you guys have a question? Sorry. What I heard somebody say. Again, this might not be something you learned. Make a guess. Because we're going to talk about that in chapter 7. Yeah, good job, guys. They absolutely can. Bacteria are cells. They're individual single cell species. Any cellular organism made of multicellular or single cells. Anything that's a cell can be infected by a virus. So, all right. Did you get in, by the way, Desiree? Okay. If we have to work on that in the future, we will. So, but Just be sure you sign in on that the sheet that I have up here before you leave. Because I obviously know you're here. Great. Good job. Good job. I don't know. Sorry, Brandy. I was typing this message to Brandy. Brandy can probably hear me. Brandy, if you're listening to me right here, I'm sorry if you missed the Kahoot code and you weren't able to join. If you weren't, don't worry about it too much. Like I saw you and I saw Desiree. Desiree, we mind writing Brandy underneath that with an I, B-R-A-N-D-I, so I don't forget to add her as present. In the future, we'll try to make sure that you guys, but that's just for people that didn't get in on the Kahoot. If you played the Kahoot, I will get you that way. And we'll try to make sure everybody gets on the Kahoot in the future. I know it's just the first day though, so I didn't want to stress about it too much. All right. Let's do a lecture. It's hot in here. I'm hot. I don't know about you guys. So this is just you normal person lecture. Like I'm going to put slides up. There are slides if you guys do want to follow along with the slides on Canvas. So in the modules for like week 1. It'll say chapter 1 and 3 slides. Also, at the top of each week, you will see on the modules, there is like the study sheet for week 1, like week 1 study sheet. There's a week 2 study sheet. Ultimately, there's a unit 1 study sheet. And week 1's study sheet is just that part of the study sheet that we're covering in week 1. Okay. So the unit study sheet is all of that you could take and put it into unit 1 for unit 1. And when we get to unit 1 exam, those are all the topics that I could pull from for the exam. It is not word for word questions. Like I said, I'm not big on that. I want to be sure you guys actually understand and can apply the material. So my questions tend to ask for that. I really don't have a whole lot of like word for word. If you lean a whole lot on exact wordage of things, you might need to find another way to start studying. One thing that I suggested to some people, because I know all of you love a Quizlet and you love a flashcard. Okay. It has its place. I'm not poo-poo on it totally. It has its place. If you guys are one of those people that you find the comfort in that because you've been doing it for so long, I highly encourage you when you make your flashcards or Kahoots, not Kahoot, well, you can use Kahoots, but you know, in Quizlets. You do flashcards or Quizlets, reword it in your own words. Okay. All right. You can start off with it in just like basic literal word for word, but as long as you at some point are rewording it, rethink about it. Don't just rely on exact verbiage. All right. All right. So let's get into this. Like I said, I will pull all of my questions for the exams, which are all multiple choice, by the way. I'm never gonna make you spell Escherichia coli or, you know, whatever. But you are gonna have to recognize what the right names are. I am not going to, there's only one question that you're gonna see that's gonna be like one of those things where it's like, what's the one that is like the exact right formatting and spelling, okay? And that's gonna be dealing with when we get into binomial nomenclature and how to write the names correctly. We'll have one question on the test like that. And it'll be which of the following bacterial names are written correctly according to the rules of binomial nomenclature, which we'll talk about in a moment. So that's the only time you're going to see that. Other than that, I'm not going to give you Escherichia coli spelled 5 different ways and you have to pick the right spelling. That's the stupidest, right? Because whenever you're in nursing school, or you're an actual nurse, the fact that you know that your patient has E. coli is enough for you. You don't need to know how to spell the freaking name exactly. And then other people, I know anatomy is more picky about that. And like when you're talking about medications, like when you get into pharmacology, that might be a more picky thing. But for your concepts for intro to micro, I'm not as concerned. Okay. And take it up with, if you like feel like that's a really important thing to you, go take it up with my boss, Dr. Rhodes. I know y'all like him anyway. If you don't know what I'm talking about, you ask somebody. So the microbiology. Microbiology is basically the science of things that are too small to be seen with the naked eye. So you have to magnify it to some degree. Even when we're talking about like parasites, like our worm parasites, our parasitic worms, like tapeworms, for example. You can see a tapeworm with your naked eye, right? But most of the time, those things are like, when you have them, they're deep inside of your body most of the time to where you wouldn't know. So, as you're in vet school, dogs that have heartworms. Do you see the heartworms typically? No, right? You have to do a blood test to look for like the larvae in the bloodstream or something, right? And so those are microscopic. So you might be looking at something like that. Fecal tests to look for the eggs and the larvae of these organisms, things like that, right? So those still at the microscopic scale, even whenever these might be macroscopic organisms. Those parasitic worms, by the way, are animals. They belong to the kingdom Animalia. That's kind of creepy to think about, right? These are the types of pathogens that we will be discussing in the class, like our friends, like heartworms. I adopted 2 dogs 1 year ago. You guys might have seen it on like the if you've already opened your stuff, you've already taken the lockdown browser quiz, and then you've already answered the question where I actually have 3 dogs. I have 3 dogs. The 2 that I adopted a year ago were Spaghetti and Meatball, and we named them. Their names were Blackie and White Sox. Like, no, right? So their names are Spaghetti and Meatball now, and they had heartworms. And so we had to put them on ivermectin and keep them kenneled for like an entire like 6 weeks. They could only go out on leashes because as the worms die, they could get stuck in their lungs and they're causing embolism and things like that. So parasites, pretty serious issue, especially with pets. Can people get parasites? Of course they can. Do people in the US get parasites? Of course they do. If you guys have kids, anybody have kids in here? Yep. Anybody ever heard of pinworms? Yeah. Everybody's worked in like GP practice or worked in an ER or had little kids, you've probably heard of pinworms. It's when little kids' booty holes get itchy, okay, and the doctor will be like, you should put tape on the booty hole overnight and then take it off in the morning and see if there's worms on it. That's legit what they'll tell you to do. And you can get treatment for it over the counter. Typically, if a kid has pinworms, they'll treat the whole family because you probably have it too. You probably just kind of like lice, okay? So it's like, you probably got it too. Yeah, we still have that here in the United States. Pretty easy to get, playing in like sandboxes at like parks and stuff like that don't have like the more sanitary conditions. But yeah, it's still prevalent. Obviously, you've heard of tapeworms and things like that. Protozoa, these are single-celled organisms. We'll talk about, let's like malaria as an example of that. Fungi, this includes molds and yeast. Prokaryotes. So this is a category. We'll talk about what a prokaryote is in a second. That includes bacteria and archaea. Most of the category, most time when we're talking about prokaryotes, we're talking about bacteria in this course. Archaea, they just don't do a lot besides live at like deep sea vents and like in like boiling springs and those like weird, super extreme environments, which don't really apply a lot to medicine. So we won't talk about archaea a ton. Too bad there aren't any dental people in here. If you guys were in here, you saw the girl that came say hi to me. She's actually a dental hygiene student that I had last semester. Like, you get them few and far between, but we will talk a little bit about oral health. We're just gonna make everybody want to brush their teeth. One of the main contributors to serious like gingivitis and stuff like that is an archaea, and it's like the only time we're going to talk about archaea. So then we have the acellular, what you might consider non-living. Are they alive? I don't know. I mean, science is like not sure about that. But this viruses and prions. Pretty little accurate picture of what viruses that infect bacteria look like, actually. So neat. Prion is just a misfolded protein. We'll learn about those in chapter 7, okay? So we talked about prokaryotes. There's basically 2 groups of cellular organisms. So a cell is a discrete unit of life that is membrane bound. It always has a membrane of some kind. So it's just an outer layer, outer covering of some kind, that has everything contained in it that would allow it to replicate itself, to make a copy of itself. You guys have probably heard about this through like mitosis. That's probably not a new word for a lot of us in here. That's basically whenever like one of your cells makes a copy of itself. You skin your knee and you need to heal that up. Your cells are just making copies of themselves through mitosis, right? The fact that a cell can beget a whole other cell that could still yet beget another whole cell, that is what we define as life. It can do that on its own. That's what cell is, membrane bound thing that could have everything in it that needs to make a copy of itself. The two types of cells that we know of in biology. That's going to be our pro or eukaryote. They're switching the freaking terminology, which makes it nice and easy for you. Okay? It's either eukaryote or prokaryote. They're the same thing. And then the other category is eukaryote. So nice. So we have some words here we can break down in a little bit. Eukaryote, like asymptomatic. When you're asymptomatic, it's without symptoms, right? If you might be sick with something, but you don't have the symptoms. A third of people that are positive for COVID are asymptomatic. How about that number, right? They're spreading it around just like the sick people are, but they're asymptomatic, right? Eukaryote means without karyote. So what does that mean? So that means nucleus, okay? Have you ever heard of a karyotype? You may or may not have. It's like when they take all your chromosomes and line them up and count all pairs of 20, 30. That's a karyotype. Just looking at what's in your nucleus. So karyo means nucleus. So eukaryote means without nucleus. Prokaryote means before. Like if you're proactive, you want to get paired before. And prokaryote, before nucleus. So those are cells that do not have a true nucleus. What's a nucleus? A nucleus is literally just a structure in our cells because we're eukaryotes. U means true. Okay? So a nucleus is a membrane-bound structure, another membrane within a membrane. A membrane-bound structure inside of your cell that just contains your genetic makeup. Right? So it's like the Smithsonian Institution. Everything, all of the information you could ever need for your entire cell to encode an entire whole new you is in every single nucleated cell in your body. Your red blood cells don't have nuclei. Or take just your red blood cells from your blood, only your red blood cells, and that was found at the crime scene, just the red blood cells. Couldn't identify the killer. There's no DNA in them. And that sounds fun. You'll learn this thing. So, anyways, so the other cells in your blood do though. So don't go leaving your blood everywhere and thinking you're getting away with something. Because your white blood cells will make up for that, believe me. So anyways, any cell in your body that has a nucleus, it has all of the information to make a whole new you. There's not different genetic information in your skin cells versus your liver. We're just turning on different genes in those areas with different specific signals. And of course, you're going to learn as we go through the course, what are those signals most of the time? Proteins, right? Just different proteins telling your body what to do. And what's all that boil down to? Chemistry, don't you love it? So all of life is dependent on that chemistry. Anyways, coming back to the point, the nucleus is like the Smithsonian Institution. It has all of the info you could ever possibly need, but you're not going to go in there and check out the whole Smithsonian Institution every time you need to build a chair, right? You're going to go in there and you're going to copy down the instructions for the chair and then take that with you home and wherever else you need to do that. So the nucleus is like that. It houses all of your genetic information. We'll come in there and make copies of the genes that we need and then work with them outside of the nucleus. So it's like that safe house, that control center where everything's contained and nice, safe place. In bacteria and archaea, those are our two prokaryotes, or eukaryotes, whichever term. I love those too. Anyways, the bacteria and the archaea are eukaryotes. They don't have a nucleus. All of everything that they do in their cell is just floating around in their goo that we call cytoplasm. It just all happens in there. They don't have anything defined by any membranes. You guys have heard, we know in the nucleus and that it contains our genetic info, our DNA, right? You guys have heard of a mitochondria probably, or an endoplasmic reticulum. We'll talk about what those do later on, but those are all membrane-bound structures that we call organelles. So tiny organs, that's what the L is for, tiny little organs. The membrane-bound structures in your cell, bacteria don't have those. It all just happens in the goo. So if it needs a membrane to be like structurally supportive of what they're doing, they just do it on their membrane, their cell membrane. They just do it there. So way more primitive. It also means they can be a lot smaller. We'll talk about that too. So those are the big differences. The big take-home message here. It's in the name. They don't have a nucleus. Okay. Let's talk about these guys and what they kind of do for the world. So about 70% of Earth's atmosphere, what we consider to be the oxygen that we breathe in our atmosphere, comes from microbes. In fact, there's heavy scientific evidence that suggests that our atmosphere lacked oxygen completely. Then life evolved. However you want to think that happened, I don't care. That's on you. But somehow life came about, okay? And then that life existed without oxygen. But they made oxygen as a byproduct once they started seeing the sun. And that byproduct of oxygen, other life evolved to use that oxygen to make energy. And we'll talk about how that works later on in other chapters. But microbes did that. And the ones that made that oxygen as their byproduct are the ones that put oxygen in the air to begin with. They're the first ones to put it in their air. And the whole reason that we even have oxygen today is thanks to that foundation they built for us. And even today, they still contribute about 70% of our oxygen content. Most of these guys are algae. Algae are not plants. They are protista, and we'll talk about that. Not much, because that's not medical, and I'm not that interested in that side of it, but a little bit. Decomposition, they're also important for that. If you guys need me to tell you that, then I don't know what to say. But yeah, bacteria and fungi are the guys breaking down the dead things. We also use our microbes. Obviously, we know we put yeast in like our bread and to make our beer. They ferment, right? We're going to talk about what fermentation is and why they do it in other chapters as well. So we'll talk about that, but we know we can utilize their products from fermentation, whether that's acid, like we get in our yogurt or our kimchi or kombucha, gives it that acidic taste, or the alcohol that comes like in our beer. You drink beer and it's got bubbles in it. Most of us know that's carbon dioxide, right? You know, they don't put that in there. The bacteria make that. It's pretty neat to think about, like also gross when you think about it, but yes, that's where that comes from in like fermented products. That carbon dioxide coming from fermentation. So it's pretty neat. We have those products that are made from fermentation. We'll talk about how that happens in other chapters. Also biotechnology. We've completely hijacked the simplicity of these organisms and put them to work for us, right? So we can take the simplicity and the ease. These guys multiply so fast. Just like within hours. When I worked at a food, so I came from industry before I came to teach here. And by the way, I'm not Dr. Yost. I'm Professor Yost. I don't have my doctorate. I did go to graduate school to get my doctorate, and I dropped out for a guy. That's a story for another time. Okay, but I do have my master's degree because I made it through a good portion of my PhD. But anyways, I moved into industry after that before I came to teach. I worked in a food safety laboratory, and we did all the testing for like McDonald's in the Oklahoma area. And anyways, you put some beef patties into some basically sterile broth and incubate it in body temperature. And then like 9 hours later, it's like completely overgrown with E. coli, if it's there, right? Yeah, it's fast. Yes. I will tell you guys. Right. I'll tell you guys about like kind of my story and everything like that when we get, like I usually say those for test days. It's kind of like a diffuser before we start tests. So I'll tell you a little bit about my personal life when we have time. Yeah, when we have time. I'll tell you this, that McDonald's is easily the cleanest place that we ever tested. Like yes. Very, shouldn't be too surprising. They're so like mechanical and like they are a machine, man. They have everything down pat and how they work everything. So way less likely. I'm not going to say we didn't test everywhere, right? We tested the places that hired my company, but McDonald's almost never failed. And there were other places that were way more likely to fail. Restaurants that are like actual proper restaurants were more likely to fail than fast food chains by far. So yeah, so put that in perspective. That's another discussion. But anyways, these bacteria grow so fast is my point. Yeah. They double and they double and they double and they double, and they'll have billions after just like hours. And you'll even see in the lab, like we're leaving ours for 48 hours most of the time, and it'll just be like all cloudy where it was sterile whenever you first started. These guys multiply so fast. We can hijack that system. So instead of you have diabetes, type 1 diabetes, you're born with it and you need insulin in order to survive, let's say. You guys be dealing with patients that have this. Even animals can have diabetes, right? They can get it. If you have type 2, you might have to deal with some treatments like this. It used to be insulin. You'd have to harvest it from like pancreas from cows. We're going to slaughter the cows anyways for beef. We'll take their pancreas and extract, insulin from that and get that. That was really pretty expensive. It was kind of cost prohibitive. I mean, for sure you weren't going to get that for your pets probably. Then they took E. coli and they put a human insulin gene into E. coli. And man, those cells crank out. It's hard. And so you get a lot of insulin that they're just pumping out like it's their job. They're happy to do it. And we can purify that out. And now you've got cost-effective insulin available for everybody. Because guess what they did before they even did the cow pancreas stuff? You just died. right? You just died if you had diabetes. So it's crazy to think about the advancements. And then aside from that and all the wonderful things that we've been able to do thanks to biotechnology and that sort of stuff and bioengineering, we'll talk also about like GMOs and what makes a GMO a concern. It's probably not what you think. Okay? We'll get to that stuff. All right, pathogen. This is a term that you should probably, this is a flashcard type of term, right? So pathogen, these are just microorganisms that can cause disease. Now, a disease is any deviation from health, from perceived health, like what you think is your normal everyday life. Yeah, maybe you're a little crotchety when you get up in the mornings, and certainly, being over 40, you definitely start to see things that way. It's not like when you were in your 20s, right? I'm not jumping around like I used to. So, but things like that might not necessarily be seen as disease because it's just part of progression. But anything that deviates out of your norm could be seen as disease. Now, that's not always going to be caused by a pathogen, is it? So we know things like heart disease, stroke, and cancer. These are pretty big ones that aren't necessarily caused by pathogens. So remember that. Our big contributors here in the US are in fact going to be heart disease and cancer, things like accidents and stroke and things like that. Well after, and of course we have things like COVID. This was done, I think, in like 2022, so no big surprise that COVID was still up there on those charts. All right, we talked about how bacteria and archaea are smaller, pretty much just because they're simpler. They don't have to have those little organelles, those membrane structures in there. They don't have to house all that. They also they don't have to accommodate being a multicellular organism and the capability to do all that. So they don't have to be as big as cells. They just don't have quite as many functions either, which we'll learn a little bit about the differences as we go. Bacteria and archaea are eukaryotes. They are smaller. Our eukaryotes, they are bigger. And we have our acellular microorganisms. We're going to talk about what really viruses and prions are in chapter 7. So I don't want to get into too much detail now because I'm sure you guys are kind of a little interested in that. I mean, it's kind of interesting stuff, learning about what these are, but man, they are really just protein and DNA when you get down to it. Okay, how do we learn anything in science? It's not the first time you guys have heard of the scientific method. God help you if it is. This is basically just the way that we're going to be looking at things as scientists. You're going to ask a hypothetical question, which is our hypothesis. Hypothesis is just our guess at what we think is going on. You make an educated guess at it. You do some tests to confirm it. If it doesn't work out, then you make a new guess, and then you try to confirm that, right? That's all science is. And I'm not going to get too picky about terminology for this, like the steps in the scientific method. You guys have an idea of that now, and it's not actually a useful thing to know, turns out. Theory is just whenever you have proven time and time again that hypothesis is correct. You have done the work to show that the hypothesis is correct. Other people have confirmed it through their work that it's correct. And there hasn't been anybody really to be able to disprove that it's correct, but there might be some sort of caveat. I just realized I only have one earring in. Probably from flinging my head around the way that I do. Anyways, theory. Yes, coming back to it. I also have ADHD. I should probably mention that. So I get distracted very easily, which is why I try to have fidget spinners. So this weird thing about ADHD people is that things that other people might think are distracting actually help keep you on track. So that's what fidget spinners do for me. I know it's kind of old. Feels weird saying that's old school, but I know it's not the trend now. So whatever. That's what I do. I like it. So that's for me. Anyways, theories. There might be some caveat out there that we haven't shown yet, and they're not comfortable saying it's a law. So that's really all theory is. Let's talk about some old guys. Louis Pasteur, he disproved spontaneous generation. That's really what he did. And that's what you need to know for the test. But what is spontaneous generation? Well, back in his day, in like the 17 to 1800s, see how I don't even know what year it was that he did this? I'm not going to ask you guys what year it was. And you know what he did, okay? Everybody thought that like if you left a piece of meat out on a countertop, that meat went rancid and started growing nasty, slimy stuff on it. It got mold on it. It's got maggots on it. That's just something that things do as they get older. They would just spontaneously generate these things out of themselves. And that was spontaneous generation. Louis Pasteur wasn't so sure about this. He devised what people call it elegant, but it seems so like basic to us now. Basically, he took a sterilized broth, he boiled up some broth, something with a lot of nutrients in it, and basically left out an open container of it, and then a container that had access with air, but it would be a lot harder for what we now know as microbes to get into it. He's like, I think there's something invisible that's alive that's doing this. And he was right. The open container that was open to the air, the microbes were able to get in and grow. The one that was the nice snaky little guy that he had there, that flask where it was hard for microbes to get in, that stayed sterile. So that's disproved spontaneous generation. There is something being introduced that we didn't see that was causing this. This is obvious to us now, but we owe that concept to his experiment. There's some other guys that did some stuff too. We'll talk more about Robert Koch later and the Koch's postulates and how he came up with these steps to discover the causes of diseases. But he did that, found out a way to find out causes of diseases. Then we have Holmes and Semmelweis to thank for your doctors washing their hands. As it turned out, they noticed that the women who were giving birth at the hospital that were being handled by doctors were more likely to die than women who gave birth at home or to midwives. And they kind of walked around and studied this for a little while. And they noticed that it was because the doctors would go in and do like autopsies or handle people with like active infections and whatever, and they were never washing their hands. They didn't think they should have to. They didn't see anything on their hands except for blood. And all of us have blood and we're not sick. You know, the healthy people, like, it's not blood that makes you sick. Well, it was something that they couldn't see that was making them, they were passing it on from person to person. Man, if your doctor was digging inside a cadaver and then came and tried to pull your baby out of you without washing their hands, but that's what was happening. And midwives were washing. Well, anyways, they noticed this, Holmes and Semmelweis, and they put that into practice in medicine. We have them to thank for that. Joseph Lister took it a step further. So now we have Pasteur. You've probably heard of pasteurization. Pasteur was the guy that did the spontaneous generation stuff. But Lister, we have his name on Listerine. Yeah, he's that guy. Okay. So cleaning solution. Now we use it for our mouth, but he found a cleaning solution called carbolic acid, which is also known as phenol. And he applied it to his surgical suites before he would do surgeries, and noticed that people were less likely to die after surgery if he like cleaned things beforehand. So he involved hand washing, basically what the beginnings of what we would now look at it as like surgical asepsis. He was the first guy to start introducing that into surgery. Go figure that that would change the landscape of how diseases might affect people from these things. No, it's in the 1800s. We already talked about these guys. I don't know why there's a second slide. All right, moving on from the old guy stuff. So you do need to know Pasteur and you need to know Lister. Those guys will be on the test. But for sure in the quiz or the chapter. Nomenclature and classifying these guys. There's so many freaking bacteria out there. I bet that if I were to sit you guys down and get you guys to really think about it, you could all name quite a few bacteria, just like even general name kind of bacteria off the top of your heads. And there's just so many more that are just like nobody cares about medically, but like just might live in the soil and whatever, all that stuff. So we had to have a way to like break them down and classify them. We'll classify them based on things like whether they can ferment, what kind of environments they live in, can they stand being around oxygen, and all sorts of stuff like that. So we'll classify them based on traits. We'll also classify them based on their genetic stuff and everything like that. And then we have names for every single organism that we think counts as a species, and we'll group them in, groups that we call genera and all of that, and put them in their own little hierarchy of taxonomy. So the way we name these bacteria is usually a two-name system. So that's why binomial two-name. So when you think of E. coli, we know that E and coli are separate. And most of you guys know that the E is like an abbreviation. The E stands for Escherichia. Like I said, I'm not going to make you guys write those words out except for one time in the history of this lab, pretty much where I'm going to care about the spelling of it. And that's in a lab project when you guys submit that. I'm actually going to tell you what your you guys will confirm what your organism's name is. You'll have it on another sheet of paper. All you have to do is spell it correctly on your lab report. It'll be there spelled for you correctly. It's just be sure that you put it in your lab report correctly. You'd be surprised how many people still spell it wrong. But that's the only time. That's the only time. I'm going to make you actually like type it out for yourselves, a name of one of these organisms correctly where it's going to count for something, okay? And even then, you might miss 1 point out of 100 if you misspell it, okay? That's how much I care. And anyway, but you have to know for the first exam, for Unit 1 exam, I will, like I said, give you a question that might say something like, please choose the organism name that, best fits the binomial nomenclature rules. And there will be some that are italicized, some that follow the rules of capitalization of the first word, lowercase the second word, and that sort of stuff. So if we are typing a name out in binomial nomenclature, it must be italicized. If you're writing it by hand, you should underline it. So that's that rule. And then the other rule with it is that the first part of the name, like the E in the E. coli, will have to be capitalized. The second part of the name, like the coli, that's going to be lowercase. Okay. Cool. So this things like E. coli, or like down here we have Staphylococcus aureus. So Staphylococcus, capital S, aureus, little a, and it's all italicized, binomial. Okay. So Staphylococcus aureus, that's staph, what you might think of with MRSA, which is methicillin-resistant staph aureus. So that's binomial nomenclature. And we abbreviate it because these names, y'all. Okay. All right. So there's taxonomy. You've probably seen this in like 8th grade science or something. Back again. So you do need to know this for the Unit 1 exam. It'll never come up again. So know it in order for the Unit 1 exam. Kingdom, phylum, class, order, family, genus, species. It's one of those ones that even I don't know it. Like if you were to be like, oh, which one comes before order? Professor Yost, I'd be like, well, I have to sit there and think to myself, kingdom, phylum, class, order, family, genus, species. I would write it down and I'd be like, oh, looks like file, looks like, class, even though I had to look at the thing. I had to write it down, y'all. I'm not even kidding. So I don't care about this, but you will have a question on the test because I have to test you over it. Okay. They say so. I don't get to make that rule. So we have domain at the very top. We all know kingdom, phylum, class, order, family, genus, species. King Philip came over for some good spaghetti. That's one way you can think of it. Make whatever acronym you want to remember it. Domain is up about all of them. We've actually already learned about the domains. There's only 3 of them. We learned about cells. Anything that has a cell, membrane bound, can divide itself, make a copy of itself, right? And we found out there's 2 kinds of cells. The ones that have a nucleus and the ones that don't. Those are the eukaryotes and then the akaryotes, right? And then within the akaryotes, there's 2 groups, bacteria and archaea. So that's our domains. There's 3 domains, eukarya, bacteria, archaea. So we kind of already know those guys. This is how they're kind of divided on the evolutionary tree. There was something that existed here, and one ancestor became a bacteria, and one ancestor branched off and became archaea, and eventually eukarya. So archaea, like you look at them in a microscope and you think they were bacteria. You do a chemical analysis of them, you think they were aliens. You look at their ribosomes alone, we'll talk about what ribosomes are. If you don't know, that's okay, we'll get to them. They're a structure in the cell that helps with making proteins. Our ribosomes and archaea ribosomes are the same, completely different for bacteria. So it's like they're a weird mishmash, okay? All right, chapter 3. We're gonna start off with our tools for microbiology, kind of how we're gonna study these guys. These are the 5 I's. We're gonna try to crank through this. We're going to talk about each of these 5 I's. This is a great infographic to come back to when you guys are studying, okay? We're going to go through each of them individually. The first of the 5 I's is inoculation. Inoculation just means we're introducing microbes to a sterile culture. So it's like a sterile media. I have completely bubbled and boiled over my broth. It's sterile now. And now I can swab your throat, put it in that sterile broth, and put it at body temperature and let it sit and see what grows. That's inoculation, is introducing that swab into that sterile. So inoculation, taking a sterile media of some kind and putting a sample in. Right? Sterile means it's void of all life. In case that wasn't clear, I think most people know that. We have different kinds of media that we use. The big kind of states they exist in is solid and liquid, and you can have variations in the middle there, gray area. You can have chemically defined and complex. We'll talk about those in a minute. And we have different functional types. We'll talk about the different functional types as well. Most of them kind of speak for themselves. So, liquid media obviously is in a liquid state. I don't really feel like I need to go over this stuff too much with you guys, but semi-solid, It has almost like a jello-like consistency. It has softness. And then we have solid media. It's not hard like this, right? But it will have softness to it, moisture for the organism, as well as the nutrients it needs, but like a nice smooth surface where we can start forming colonies. We'll talk about what those are. The way that we give media that sort of solid aspect to it, that jelly-like consistency, is by adding agar. It's just a sugar that we get from algae and makes it solid. Right, we have defined and we have complex media. This defined is very chemically, you know exactly what's in it. You literally put in 5% sodium chloride and then 3% of this exact amino acid and whatever else you're adding in. You know, be defined. You know exactly what chemical, what composition it's in. So that might seem like, wouldn't that be all the media that you're making though? It's very useful in research because you know exactly what nutrients and at what concentrations. However, actually most of the time that we use a lot of media, it might fall under complex. So one of the things that we use very commonly might be blood agar. It's just like nutrient agar that we added 5% sheep's blood to. Do we know what's in every single sheep's blood to be? No, we don't. So that can vary, but we know it has sheep's blood in it. So that's what we mean by complex. All right, the uses for these media. General purpose, I feel like that speaks for itself, but enrich. Let me ask you guys something. You guys know that if you go without vitamin C in your diet, you'll end up with scurvy. I mean, you might not know that, but hey, newsflash. You have to get vitamin C in your diet. Or if you don't get any vitamin C, like the sailors of old who used to just eat like, hardtack, if they didn't get limes and stuff like that to add vitamin C to their diet, they would get scurvy. You can't make vitamin C. No matter how hard you might think about it, no matter how hard your little cells might try, they can't make it. They don't have the ability to make vitamin C. So it has to be supplied in your diet. You actually have a lot of things like that that you're probably some of them aware of, like B12, a pretty common one for women. They don't absorb enough of it in their diet a lot of times and have to supplement that, or iron, or things like that. Like these are things that your body can't make for itself that we get from our diet. So those things, we call those growth factors. Bacteria have some stuff like that, depends on the species. Okay, if bacteria needs a growth factor added in its media, then that bacteria is called fastidious. And the media that it would grow on that has the growth factor in it is called enriched. So those are the three terms that kind of go together with that concept. Okay, be aware of that. Next, we have selective media. This is media that has something in it that inhibits the growth of other organisms. There's two major groups of bacteria we're going to talk about in chapter 4. Don't worry about what they mean right now, but they're going to come up so often that I swear by the end of this semester, you're not even going to think twice about them. Gram-positive and gram-negative bacteria. Okay, so we have lots of media out there that will allow growth of just gram-positives or growth of just gram-negatives. We'll add something to it that inhibits the growth of the other. Things like strep throat, that's a gram-positive bacteria. You don't want, if you're trying to figure out, you're pretty sure that your patient has some sort of gram-positive strep throat type bacteria, then you might not want any gram-negatives interfering in the growth. So you'll use something that selects for that to culture it. Or you're not sure. Maybe this is a gram-negative thing. I mean, you can't do a gram stain for some weird reason. Then you might just slick it on some media and see if it grows or not, and that could tell you. So these guys will usually select one group to grow and inhibit the other. Whereas differential media, anything can grow on it. It's just some things might change in a different color, usually a color change. And it's always going to be related to some sort of metabolic byproduct. Things like when something ferments something, it makes acid, that causes a pH change, and there's pH color indicators that'll change color, stuff like that. So you can imagine there will be media that you could make that selects only for gram-positives to grow and has a pH indicator in it. So it would be both selective and differential. Cool. One type of differential media that there is blood agar. We've already mentioned it. It might have sheep's blood in it. A lot of bacteria, like the pathogens that grow in your body, might be able to break down your red blood cells. That's called hemolysis. So hemolysis, hemo being blood, lysis being breakdown. So if you can break down red blood cells, there's different categories of this. There's 3 kinds of hemolysis. Okay, one of them is don't ask why. Okay, but one of them is it doesn't cause hemolysis. So if you can grow on blood agar and you don't cause visible hemolysis, then we call it gamma hemolysis. I don't know, okay, but that's it, gotta know it. The other two have some sort of actual hemolysis involved. Think of them as steps. The first step, we got partial of the way through. You got to get partial of the way through so you can get through all of it, right? So the first step, alpha, partial hemolysis. So you'll see like greenish color of the blood cells in the artery. Whereas beta hemolysis would be a complete breakdown. So you've gone through that second step. You've taken it further now. Beta hemolysis, we've actually got clearing of that blood cells. You can see it in this picture here. This is something that just grew on the auger where we have MCRO2124. And we have greenish in this. This is Streptococcus sanguinis. It's a common one in your mouth. If I'm swabbing and I accidentally hit your tongue, you might grow that. Okay. When you swab people's throats, you're supposed to only hit the throat. You guys know about this. You'll learn about this anyways. Anyways, if you ever had strep throat, then you know. Anyways, the bottom one there, Streptococcus pyogenes. What do you think that causes? Strep throat. So that's actual, like what you think of when you think of strep throat, Streptococcus pyogenes, and it is beta hemolytic. You can see that there. When you compare their patient A sample to that, if those were your only three choices, wouldn't it be nice? But yeah, you can see that clearly. It compares most closely to that. All right, this is just talking about how you can have both selective and deferential, which is one I kind of described to you guys. Some examples of that would be McConkey auger or mannitol salt auger. I'm not going to test you specifically over those. If I ever bring up something like mannitol salt auger on your exam, I will tell you what it is. Okay, so until you need to know that, which you will learn about what these are and have to know about them in the lab in the later part of the lab, but not for exam one. They're just examples that have salt in the mannitol salt auger and maybe like a pH indicator. They have both going on. Okay. So we talk about colonies, and we want to growing up our culture. So our steps in the 5 I's, we already talked about inoculation. Incubation was a step we kind of glazed over, but it just means putting it at the right conditions to grow. Okay. Our third step is isolation. So if you swab someone's throat, obviously you're going to pick up more than just the bacteria that's causing whatever the problem is, right? You have so many other bacteria in your mouth and in your throat and everything. How do you tell what it is that's causing the problem? So you need to isolate the organism of interest. And so the best way to do that is to get discrete colonies. Because a colony, once we get those little dots that we see on those plates, is a colony, each one of those is. If they're all by themselves and not touching anybody else, that came from one cell. And that one cell just multiplied up on itself and piled up. And so if you were able to spread out individual cells enough, you have a whole mess of them from someone's throat, but you're able to spread them out enough that you can get individual cells that will then pile up in individual colonies. You can take a sample of that with a little sterile link and put it in broth that's completely sterile, and now you have what we call a pure culture. It's a representative of just that one species. That's what a pure culture is. That's why it's useful to get individual colonies. If you swab somebody's throat, you're going to get such a mess. So how do you separate it out? That's what the streak plate method is for, and we'll do this in lab. So it'll be in practice once you get it going. It'll make a little more sense than just seeing it on the screen. But essentially, we have swabbed somebody's throat or whatever, drooged it onto a plate, and then sterilized our little loop, kind of touched it into that, spread it out some, sterilized again, it's completely clean now, zhuzhed it out some, sterilized it again, and you just zhuzh it out until we do it 4 times, it's called quad streak, and then supposedly in that 4th quadrant, you'll get these individual colonies eventually, because we spread out those cells individually, then incubated them, let them grow into the pile of that exact same cell replicated over and over again. So when you go to take it for a sample from that, you can grow your culture. Okay? So just spread them out. It's just like diluting them out, but like physically spreading them out. So there's different kinds of cultures that you would have if you like incubated this stuff up. We have mixed cultures, which is just like what you would have if you had swabbed somebody's throat. That middle picture there. Contaminated culture, maybe you got your pure culture, and then something from the air, somebody coughed on your stuff or whatever it is, went back and then contaminated. It's not mixed because it didn't start out that way in the original sample, right? So do know the distinction between mixed versus contaminated. Next, we'll do testing. I think this part's pretty straightforward too. There's a lot of tests that are out there nowadays, whether it is PCR, whether it is looking for antibodies or whether it is looking for all sorts of other things we could test for in a infection or sample or whatever it is, whether it grows or not, whether it changes the media color, whether it has the presence of DNA, whether it has all sorts of things, that falls into the inspection eye. And then gathering all that information that you get, sometimes all you need is like a rapid test, right? And you know you have COVID. What happens, like I went to the urgent care the other day because I was coughing, I had a sore throat. They did COVID. This was like a few weeks ago. COVID, flu, and strep. They were all negative. So what was it? No, wait. Yeah, it was sinus drainage. Exactly what it was. It was sinus drainage. Probably a viral thing is probably what it was. And but then I got bronchitis because the sinus drainage collects in your lungs. And hey, you know, I hate to break it to you, guys are gonna hate me for this, but I'm a smoker. I smoke cigarettes. So I'm trying to quit now because bronchitis really hit me hard. But I'm really trying, y'all. So support me wherever you can. But that it's like it really. I knew the reason that I got bronchitis was because I was a smoker and all the junk in my lungs, and my lungs weren't able to clear it out as well. So bacteria led to a secondary infection. We're going to talk all about that sort of stuff, but like what exact bacteria caused that, we would have to culture it to find out. But probably gram-positive, just because of the environment, and we'll talk about that, how you would know that. So they gave me amoxicillin, which is good for gram-positives, but I will probably never know the exact microbial cause of the actual bronchitis part of it. Testing can tell us that stuff if we need to know it. And sometimes you might. If I was blue in the face and I'd already taken amoxicillin and it wasn't getting better, you might want to culture it and figure out what it is to better target it. We kind of already talked about the size of things. I'm not going to get into it too much, but it's part of the reason why we need microscopes. We're going to finish this up real quick, I promise. Microscopes, we use these to see all these organisms. You do need to understand how a microscope works for the lecture. And there will be questions about it on the lecture, but we're going to talk about it in enough detail in the lab. I'm not going to cover it really in detail, okay? You're going to know the parts, the basic parts of a microscope and how they function. The fact that when light comes out of the light bulb, it gets focused through your specimen and hits the first lens that magnifies it. That first lens is the one that rotates. The bottom is the objective lens. You have a lot of different powers of lenses that magnify at different strengths. So that's the objective lens. That's the one that creates the first image, which is the real image. Yes. Yeah. So a lot of people, people ask me about this all the time. I don't even think about the fact that I really only look through one eye. So yeah, a lot of people really struggle to try to see through both eyes. Don't worry about it. As long as you're seeing through one eye, you'll get the information that you need and you'll get more comfortable with it. We're going to use microscopes every day. So you'll get so used to it and so good at it. Hopefully you'll get good at it anyways. Yeah, that you'll get better at it and you won't even think about it. Yeah. Yeah, everybody else is looking with two eyes. Maybe I'm the only one guy. It's normal to only use one eye. If you can look with both two eyes, congratulations, I guess. I don't know. Sure. We have plenty of time to get practicing on that. Anyways, so big properties of a microscope are going to be magnification, resolution, and contrast. Contrast, of course, is going to be like the difference between saying light gray and dark gray versus black and white. So really getting that definition there. When you look at a cell is made of like pretty much almost clear stuff. So it's really hard to see cells if you don't stain them, which is why we'll do stains. So we have the objective lens where the light hits first, then it comes up through mirrors and goes through another lens before it hits your eye. That lens is called the ocular lens. So there's another magnification going on there. So the first image is made by your objective lens through that specimen. That's the real image because it's the first one that's made. The second one gets made again through that second lens before it hits your eye. So that's the virtual image. Think of virtual as being like visual. Similar word. So that's where your ocular lens would be. That's virtual ocular. Visual. We already know what magnification is. Whoa. Okay, if you're trying to calculate the magnifying power overall of your microscope, like we said, there's two lenses. The one that's on the bottom that rotates and when you're looking at. All the ones in our microscopes are 10x. This is where I tell you guys you'll have to multiply by 10. So you'll have your, whatever your lens is on your objective lens, 4, 10, 40, or 100, multiplied by whatever your ocular lens is, which is 10. Okay, so if you're at the 100x lens and you're at the 10 up here, 1000x. Okay. Pretty easy. Refraction, when light goes like from our light bulbs, like up here, from the glass that's in like a light bulb and it hits air, it diffuses out, it refracts, and it goes everywhere. And that's how we light rooms. It's great. But you don't want that when you're looking at your specimen because you're losing detail when you lose those light rays. So what happens is we want to keep that all in one place. We want it all getting to our eye so we get as much information as we can. We don't want it lost in the air. So going from glass to air is a big change. So we want to minimize that change as much as possible. Kind of already talked about contrast. And so this is getting into oil immersion. So we use oil to mimic the glass as much as possible. It helps contain, you see the picture here, we're containing our light rays within the oil so they don't scatter out, contain as much detail as possible. So every time we use our 100X, we will be using oil immersion. We're almost done, I promise. So when you're trying to look at your organisms, you'll be looking at them based on whatever you're trying to analyze. If you want to look at your organism while it's alive and swimming, you don't want to kill it, right? So you would use something like maybe a wet mount for that, where you have actual broth and living organisms. The problem there is that it can dry out pretty quickly, as well as you can't really stain them to get contrast. So that can be a problem too. And it's, you risk contamination because you're live organisms, right? Heat fix is something where we will take the sample, dry it out, pass it through some flame. That makes sure everything's stuck to the glass where it should stay. So that when we do all our rinses with our stains, it stays in place. It kills the organism, so you're not doing something contaminated. It also keeps all the parts that might be in the cell also in place, so that you can look at that stuff too, really reliably. That's what heat fixing is for. A lot of times our stains will be dealing with that. There's a few kinds of stains, basic and acidic. So I think a B looks more like a P. And if you look at the capital N, it kind of looks like a G to me. This is how I remember these. These are my mnemonics on how I remember these things, okay? So basic are cationic, or they have a positive charge. That positive charge is attracted to the negative charge of your cells. Just believe me when I tell you that they're negative for now until we get into the chemistry of things, okay? Cell surface is negative thanks to those phosphate groups on the outside of your membranes. So the positive is attracted, opposites attract. So it sticks to the surface of the cells. However, acidic, negative, they are repelled, just like South Pole to South Pole magnets. So it'll stay in the background, not the cells. There's some pictures of that. A lot says the same thing. You can see here the cells are stained on this one for a positive stain, opposites attracted. Similars repelled one another. So negative repelled negative, and we stain the background instead. Simple stains, we're just using one stain just to stain to get contrast typically. Whereas differential stains, we're trying to see different structures within the cell. Sometimes we'll be staining just some structure that only some cells have. So when you rinse away that stain, if you didn't have it, you're not stained anymore. So you have to counter stain with another color. I'm not gonna lie, like most of the stains that we're doing in the first half of the class are gonna be differential stains that are kind of like that. A lot of counter stain, just to help you just see the cells if they were negative. Okay, that's the idea. We'll talk about the different kinds of stains when we get into chapter 4. All right, so we're gonna go over the questions since I know we're done with class. What is a pathogen? This is where normally we would go in order. So this would have been Lakita's question, for example, right? What is a pathogen? A pathogen is a microbe that causes disease. I went the wrong way. Can you name one non-cellular pathogen? So you could have said virus, or you could have said literally like COVID or flu. Those are viruses, right? The 5 I's, that was a little bit more involved question. You have inoculation, incubation, we have isolation, we have inspection, and we have identification. There's just steps that you go through to try to ID whatever your organism is. Selective media is a type of media that you would select for some organisms and others would not be able to grow. Whereas, remember, differential, it's just gonna have a color change, but anything could grow on it. It's just a matter of whether or not it makes the product that causes the color change. And what part of the microscope generates the real image? That is the objective lens. So that's it for today. On Thursday, we do have lab again. We're just gonna be talking about like the class structure and note taking and like what to expect in lab and things like that. Just general overview stuff we didn't have time for in lecture. You don't need your lab coat on Thursday, but starting on week 2, please have your lab coats, okay? There are not lab materials. A lot of people come to me, they're like, I don't have the lab manual. You are correct, you don't have the lab manual, or rather, you're incorrect because you do, because it's in a PDF document on the Canvas page. So I think it's a waste of your money to make you buy a lab manual that you're never going to use again after this class. So I've made my own lab manual. I've published it in a PDF format on Canvas for you guys. You do not need to print those out to bring to class unless you want to. I will provide all the lab manuals in class. They stay in class, the physical formats of them do. And they are in sheet protectors, and we will write on them with dry erase markers. You will take a picture of them and upload them to your assignments. If you prefer to write on your own paper that you have, like you're welcome to print out your own, but I will not be printing them for you, and you're not required to. That would just be a preference thing. Other than that, you're more than welcome just to use the physical ones that we have for the dry erase in lab. Cool? Yes. Correct. Right. I just want to keep you guys on the same schedule as my Monday, Wednesday. If like that's what I don't like about it, right? It's like if they get off on Monday, you guys get a whole extra day of class compared to them. You guys think about that ever? Because I think about it a lot. That's what I do is try to schedule this stuff. Oh, what? Why would we? So no, we do not have class on Tuesday. Just keep you guys on the same schedule as Monday, Wednesday. Labs are Tuesdays and Thursdays immediately after. Yes. Yes. 7:15. So this lecture goes to 7, and then lab is 7:15 to 8:30. We'll start lab this Thursday, but it's not really lab. So you'll have to be there, so required attendance. We're just going to talk about class stuff and what lab will start next week. It'll be Tuesday and Thursday on, right? Don't we love it? Week 3, we'll start actual Tuesday. Yeah. Yes. Starting on week 3, it will actually be Tuesdays and Thursdays. Make sense? Lectures both days, labs both days, starting week 3. I know it's weird because of the Martin Luther King Day. I appreciate it. Yeah. All right. Anybody have any other questions? All right. Huh? Yes. Every Friday. I think I mentioned it in the email, but I don't remember if I did. Every Friday at 8 a.m. you'll get an email from me telling you that by midnight on Friday you have all of these things due. So every Friday by midnight you will have whatever the week's things were. So this week you will have chapters 1, 2, and 3 quiz due by midnight on Friday. 1, 2, and 3 quiz. Chapters 1, 2, and 3. Are they individual? Yes. And they'll be on your to-do list. Like I know a lot of classes, like I don't know for sure, but from what I hear is that chemistry is like things will open up as we go through the class. Not my class. My class, everything is open. So everything that you see that's going to be due eventually in my class, it's already up. Like you're not going to have other things popping up as you go. So everything's already up. I don't need to do it. You already signed into it. Yeah. Yeah, like surprise homework.
Yes. I thought it was only Thursday because it says TR.
Yeah, TR, yeah.
But I came to buy a book and that's why I asked them. They're like, yeah, you have.
Yeah, by the way, you're not the only one. That happens at least once every semester. So, yeah. I hope they change that. Like, I hope they make that clearer.
Like T8 or something.
Something, yeah. Yep.
Lab coats, they can be like the short ones. We don't need like fancy ones.
Right, and I don't know what color they are either. Yeah.
Well, I have one from the nursing program and I'm not buying another.
You're fine. Yeah, that's fine. Yep. All right. I don't know if you're still there, Brandy, but I'm gonna cut you off now.
When you were talking about media, you're talking about like the environment that you place microbes in to